ribosomes, mitochondria, plastids, and endoplasmic reticulum. I am some teach you about it for four topics and one cell organelles. But first of all, if you are new, so please subscribe my channel, press the bell icon, and you start my topic to discuss about it. So now I am start. Cell organelles are small structures within cells that perform dedicated functions. There are about a dozen types of organelles commonly found in eukaryotic cells. We will go through the basic facts about important cell organelles and its ribosomes. Ribosomes are tiny granular structures that are either floating freely in cytoplasm or are bound to endoplasmic reticulum (ER). Each ribosome is made up of almost equal amounts of protein and ribosomal RNA. And rRNA ribosomes are not bound by membranes and so are also found in prokaryotes. And eukaryotic ribosomes are slightly larger than prokaryotic ones. Ribosomes are the sites of protein synthesis. Protein synthesis is extremely important to cells, and so large numbers of ribosomes are found throughout the cells. And when the ribosome is not working, ribosome cells is not working, it disassembles into two smaller units. And mitochondria are double membrane bonded structures found only in eukaryotes. They are sites of aerobic respiration and are the major energy production centers. The outer membrane of a mitochondrion is smooth, but the inner membrane forms many enfoldings called crista and singular crista in the inner mitochondrial matrix. This serves to increase the surface area of inner membrane on which membrane bound reactions can take place. And in mitochondria have their own DNA and ribosomes. The ribosomes of mitochondria are more similar to bacterial ribosomes than to eukaryotic ribosomes. And plastids information. Plastids are also membrane bound and organelles that only occur in cells of plants. And photosynthetic proteins is included in algae. There are three more types: are chloroplasts, leucoplasts, and chromoplasts. Like mitochondria, chloroplast information. Chloroplast is also bound by a double membrane, and the outer membrane is smooth, while the inner membrane gives rise to sacs called thylakoids. The stake of thylakoids is called granum, and grana float in the inner fluid of chloroplast, stroma. Chloroplasts are the sites of photosynthesis in eukaryotes. They contain chlorophyll, the green pigment necessary for photosynthesis, and such associated pigments. These pigments are present in the thylakoids of grana. And the second type of plastids in plant cells are chromoplasts. They contain pigments associated with bright colors and are present in the cells of flowers, petals, and folioles. Their function is to give colors to these parts and thus help in pollination and dispersal of fruit. And leucoplasts Leucoplasts are the third type of plastids, and they are colorless and store starch, proteins, and lipids, and they are present in the cells of those parts where food is stored. And endoplasmic reticulum, ER. Endoplasmic reticulum is a network of interconnected chemicals channels that extends from cell membrane to nuclear envelope. The network exists in two forms: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is so named because of its rough appearance due to numerous ribosomes that are attached to it. Due to the presence of ribosomes, RER serves a function in protein synthesis. And smooth endoplasmic reticulum SER lacks ribosomes and is involved in lipid metabolism and in the transport of materials from one part of cell to other. It also detoxifies the harmful chemicals that have entered the cell. And Golgi apparatus. Golgi apparatus, an Italian physician, Camillo Golgi discovered a set of latent sacs or cisternae in cell. In this set, many cisternae are stacked over each other, and the complete set of cisternae is called Golgi apparatus or Golgi complex. It is found in both plant and animal cells. It modifies molecules coming from rough ER and packs them into small membrane bound. Small membrane bound sacs called Golgi vessels, and these sacs can be transported to various locations in cells or to its exterior in the form of secretions. And lysosomes. In the mid-20th century, a Belgian scientist, Christian Denis de Dieu, discovered lysosomes. They are single membrane bound organelles. Lysosomes contain strong digestive enzymes and work for the breakdown digestion of food and waste materials within cell. During its function, a lysosome fuses with the vacuole that contains the targeted material, and its enzyme break down the materials. And centrioles. Animals and many unicellular organisms have hollow and cylindrical organelles known as centrioles. Each centriole is made of 9 triplets of microtubes and made up of tubulin protein. Animal cells have two controls located near the exterior surface of nucleus. The two centrioles are collectively called as centrosome. Their function is to help into formation of spindle fibers during cell division. In some cells, centrioles are involved in the formation of cilia and flagella. So my dear friends, how is my topic today about which I discuss you cell organelles and its types. If you are like it, so also please subscribe my channel, press the bell icon. So now I am go to my new best topics and give you much information for my new videos. So now I am go to my new videos. Take care. Allah Hafiz.